Greetings, students. Mr. Sanger, live from the prep room, here to talk about the bacterial transformation lab. So, the goal of this lab, you're going to carry out transformation of a plasmid into bacterial cells. Hopefully you know by now, transformation is where bacterial cells take in a random bit of DNA from the environment. So our goal is to try and get it to take in a particular piece of DNA that we want to give it. You're going to use what's called a selectable marker to determine which bacterial cells took in the plasmid. And then 3, you're going to measure the rate of transformation in the lab. So basically, the way it's going to work is you are going to get live bacterial cells, and you are going to expose them to a plasmid and determine if transformation occurred. Now, maybe you remember this, maybe you don't, but let's just recap it. DNA do not have bacteria do not have a nucleus, but rather have this single circular chromosome. But bacterial cells can also have these little extra rings of DNA called plasmids. And so in this lab, we're going to basically take a genetically engineered plasmid, and we're going to try and induce this bacterial cell to take that genetically engineered plasmid inside of it, and then we're going to look for it later on. So let's talk a little bit about that plasmid that we're going to introduce. Here's a little model of what it looks like. Some features. First of all, it has an origin of replication. Remember, DNA replication always begins at an origin of replication. So this is going to ensure that the plasmid is able to be copied as bacterial cells divide. Second, it's going to have a BLA gene. I like to call it the BLA gene. And this gene gives it resistance to an antibiotic called ampicillin. So antibiotics kill bacteria. So ampicillin is basically a drug that is used to kill bacteria. It's a medicine, actually. But this gene is going to make these bacterial cells resistant to that antibiotic so that it doesn't kill them. It also contains the GFP and ERA-C genes. Together, these protein products can cause green fluorescence in the presence of UV light, but only when arabinose is present. So let's explore that a little bit further. So the ERA-C gene codes for an arabinose-dependent inducer of GFP expression, basically this guy. It codes for this little protein. Now, and then GFP codes for this green fluorescent protein as down there. Now, the natural situation, the way this works in the natural world under normal circumstances, is arabinose is a sugar and it acts as an inducer. So in the presence of arabinose, it binds to this ERA-C protein, changes its shape, which allows RNA polymerase to attach and begin transcription. And then the cell will produce these protein products that will help it produce enzymes to break arabinose down. So this is similar to the lac operon, where lactose essentially turns on the genes that are used to break down lactose. Here, arabinose turns on the genes that are used to break down arabinose. So what genetic engineers have done, though, is they've taken that plasmid and in place of these structural genes for enzymes to break down arabinose, they've replaced those with this GFP gene, which essentially makes a protein that is green fluorescent, that makes that green fluorescence. So basically the presence of arabinose will turn on the gene that makes green fluorescence. So we've just sort of replaced those structural genes with this GFP gene, okay? So here's how it's going to work. You're going to be given 4 plates. All 4 are going to be given luria broth, which is basically just food. It's bacteria food. But then 2 of them are also going to get ampicillin. 3 of them, I'm sorry, are going to get ampicillin, that antibiotic that kills bacteria. And then 1 of them will also be getting arabinose. Further, some will not have the plasmid, others will have the plasmid. And your task is to figure out what should we see if we plate bacterias on each of the above plates. So if we give it food and no plasma, what will happen? If we give it food, an antibiotic, and no plasmid, what will happen? Et cetera, et cetera. You actually have 3 choices. The first choice is no growth, meaning everything dies. A second growth option is lawn, which basically all of the kind of fuzzy, foggy stuff here is bacteria. It basically just means that the bacteria are able to grow wherever they want, uninhibited. They're doing great. The third option is colonies. So here, only certain bacteria are able to survive. Each colony is basically a visible mass of cells that descended from a single bacterial cell that was able to survive. Um, so, um, so your pre-lab task basically is to figure out, in all these situations, should we see no growth, a lawn, or just individual colonies? And I'll kind of leave it to you guys to figure out the rest. Now, how are we actually going to do this lab? So on day one, we need to what is called plate the bacteria. Basically, the bacteria come in this little vial, but we need to grow them on a big petri dish so we can get little individual colonies that look like that. We need colonies for our lab because these colonies are actively growing. And so the way you do this, and you're going to actually get to physically do this in lab, is you dip what's called an inoculating loop in the bacteria, and then you just streak back and forth about a dozen times. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Only staying in this one little section of the petri dish, okay? And you can see they did that right there. Then you, without dipping the inoculating loop in bacteria again, then you start over again and you kind of grab some of this that you already streaked and you go 1, pick up a little bit more, 2, and then avoiding overlapping this area again, 3, 4, 5, and you do about 10 more of those. And what you're doing is you're basically grabbing some of these bacteria and you're spreading them out over a larger area. You're essentially diluting them. Then you do the same thing. You go into this section once, twice, and then 3, 4, 5, 6, 7, 8, 9, 10 times, diluting it further, and then finally same thing. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and you're diluting them so much that by this point, you hopefully will have individual cells, which will later give rise to colonies. So we're going to do that on day 1. We're going to put them in an incubator, which keeps the temperature at 37 degrees Celsius, which is optimal for bacterial growth. And then hopefully on lab day, you have a starter plate that looks like this that you can get individual colonies from. So then on the actual lab day, the gist of the procedure, you'll have to follow the directions carefully, but here's kind of the gist. You're going to get these what are called microcentrifuge tubes, and you're going to label one P-glow plus and one of them P-glow minus. P-glow is the plasmid, so ultimately this one will get the plasmid, this one will not. So to start off, you're going to add what's called transformation solution to both of these tubes. Transformation solution is calcium chloride, and basically it helps make the cell wall a little bit more permeable to DNA. Remember, the cell wall is not a particularly effective barrier. The membrane is really the bigger barrier, but this does help it become even more ineffective as a barrier because the goal, remember, is to get DNA into the bacterial cell. So you're just going to add a little bit of this to each test tube, put them on ice. Then you're going to take 2 to 4 colonies from that starter plate that we did the previous day, and you're going to scoop them up in another inoculating loop and just add them in there. Okay, so at this point, you've got 2 identical microcentrifuge tubes with that calcium chloride solution and bacterial cells. The next step is where these become different. You take plasmid, which again just comes in this little vial, and you're going to add plasmid to the P-glow plus, but not to the P-glow minus. And then you're going to keep these on ice for about 10 minutes. And while they're just chilling on ice, the cell membranes of those bacteria are going to cool down. And hopefully you remember from what we've learned about membrane fluidity that the cold is going to kind of slow those molecules down and the membrane is going to kind of become packed up. Now, is that good for transformation or bad? If we want things to get into the cell, do we want the membrane to be kind of packed together and less fluid? The answer is no. So that's kind of weird, but that's going to be happening while it's on ice. So while it's sitting on ice, you're going to get 4 prepared Petri dishes, and you're going to label them with either plus P-glow or minus P-glow as indicated in the directions. Okay. So the next step is the key step. You are going to heat shock the cells. You're going to take your samples that were on ice for 10 minutes, and you're going to put them in this little styrofoam holder, and you're going to put them in a little water bath. It's like a lazy river for bacteria, and they're going to float there for I think it's 50 seconds. And that is called the heat shock. When those membranes that have been sitting on ice and are all packed together hit that hot water, they're all of a sudden going to be like, whoa, and the membranes are going to start moving. The phospholipids are going to move faster, and they're going to get really fluid, which basically makes them a little bit more leaky. And so during this heat shock, that is the key moment when we're hoping that the DNA, our plasmid, can get into the cell. After that, you're going to dump it back on ice for a little bit longer. Okay. Afterwards, you're going to take this solution and you're going to pull 100 microliters out of this and add it to here, another 100 microliters, add it to here, and then same with the Piglo minus, 100 microliters, 100 microliters, and then you're going to streak. You're just going to spread them across the Petri dish like that, and then you stack them up, tape them together, put your name on there, and then they sit in the incubator overnight. And the hope is that some bacteria manage to take in that plasmid, and that's going to affect their phenotype. And then we'll see if the predictions you made in the pre-lab were supported or denied. So that's pretty much it. We'll see you in lab, people. So long.